What are the primary functions of the cell membrane? The cell membrane serves multiple functions, including structure, communication, separation, and selective permeability. For structure, it provides physical protection and defines the boundary of the cell. For communication, it allows for cell signaling and recognition through receptor proteins. Separation involves maintaining a selective barrier between the internal and external environments of the cell. Selective permeability regulates the movement of substances in and out of the cell, maintaining the cell's electrochemical potential. Describe the structure of the cell membrane, including its components and organization. The cell membrane is composed of a phospholipid bilayer with hydrophilic phosphate heads facing outward and hydrophobic fatty acid tails facing inward. This arrangement creates a selectively permeable membrane. Embedded within this bilayer are proteins, cholesterol, and carbohydrates that contribute to its structure and function. Explain the selective permeability of the cell membrane. Selective permeability means the cell membrane controls the entry and exit of certain molecules. Small nonpolar molecules like oxygen can pass through freely, while larger or charged molecules require specific transport mechanisms such as proteins. What are the roles of hydrophilic heads and hydrophobic tails in the cell membrane? Hydrophilic heads interact with the aqueous environments inside and outside the cell, while hydrophobic tails avoid water, creating a barrier that prevents the free passage of water-soluble substances. How does the saturation of fatty acid tails affect the fluidity of the cell membrane? The cell membrane has one saturated and one unsaturated fatty acid tail. The unsaturated tail contains kinks that prevent tight packing, increasing membrane fluidity. List 6 examples of proteins found in the cell membrane and describe their functions. Enzymes catalyze chemical reactions. Transmembrane proteins span the membrane and assist in transport. Carrier proteins facilitate the movement of substances across the membrane. Receptor proteins bind to signaling molecules, triggering cellular responses. Hormone receptors specifically bind hormones to regulate processes. Adhesion proteins help cells stick to one another or to the extracellular matrix. What are the two main groups of transport mechanisms across the cell membrane? Active transport requires energy, often in the form of ATP, to move substances against a concentration gradient. Passive transport does not require energy, allowing substances to move down their concentration gradient. Define active transport and give examples of primary and secondary active transport. Active transport is the movement of substances across the membrane using energy. Primary active transport involves the direct use of ATP, such as the sodium-potassium pump. Secondary active transport uses energy from the electrochemical gradient created by primary active transport, like the sodium-glucose transporter, or SGLT. Define passive transport and give examples, including osmosis and facilitated diffusion. Passive transport is the movement of substances without energy input. Examples include simple diffusion, which is the movement of small molecules like oxygen down a concentration gradient. Facilitated diffusion, which is the movement of larger molecules like glucose through a carrier protein, and osmosis, which is the movement of water across a membrane. What is diffusion and how does it occur through the cell membrane? Diffusion is the movement of particles from an area of higher concentration to an area of lower concentration. Through the cell membrane, small non-polar molecules can move via simple diffusion, while larger or charged molecules move via facilitated diffusion using transport proteins. How does simple diffusion differ from facilitated diffusion? Simple diffusion involves the direct movement of small non-polar molecules through the lipid bilayer. Facilitated diffusion requires the use of carrier or channel proteins to move larger or polar molecules across the membrane. Explain how membrane permeability is related to lipid solubility and molecular size. Lipid-soluble molecules pass easily through the lipid bilayer, while large or water-soluble molecules cannot. Membrane permeability depends on the size and polarity of the molecule. Smaller and non-polar molecules diffuse more easily. What are the factors affecting the rate of diffusion? Can you provide the mathematical equation for this? Factors affecting diffusion include the concentration gradient, where higher gradients increase the diffusion rate, temperature, where higher temperatures increase kinetic energy and diffusion, membrane area and thickness, where larger surface areas increase diffusion and thicker membranes slow it down, and membrane permeability, where higher permeability increases diffusion. The equation for diffusion is Fick's law, J equals negative PA times the concentration difference across the membrane, where J is the flux or rate of diffusion, P is permeability, A is membrane surface area, and C1 minus C2 is the concentration difference across the membrane. What is osmosis and how does it differ from diffusion? Osmosis is the movement of water across a semi-permeable membrane from an area of low solute concentration to an area of high solute concentration. Unlike diffusion, osmosis specifically involves water. Define tonicity and explain isotonic, hypertonic, and hypertonic solutions. Tonicity refers to the ability of a solution to cause a cell to gain or lose water. It depends on solute concentration and membrane permeability. An isotonic solution has equal solute concentration on both sides, resulting in no net water movement. A hypertonic solution has a lower solute concentration outside the cell, causing water to enter the cell. A hypertonic solution has a higher solute concentration outside the cell, causing water to exit the cell. How do cells react in isotonic, hypertonic, and hypertonic environments? In an isotonic environment, cells maintain their shape. In a hypertonic environment, cells swell and may burst, a process known as lysis, as water enters. In a hypertonic environment, cells shrink, a process known as crenation, as water leaves. Vesicular transport involves the movement of substances via membrane-bound vesicles. The two main types are endocytosis and exocytosis. Endocytosis is when the cell takes in materials, while exocytosis is when the cell expels materials. Endocytosis involves the engulfing of materials. There are three types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis, also known as cell eating, is where large particles are engulfed. Pinocytosis, or cell drinking, is when the cell takes in extracellular fluid. Receptor-mediated endocytosis is when specific molecules are engulfed after binding to receptors. Exocytosis is the process where vesicles fuse with the plasma membrane to release contents such as hormones or neurotransmitters outside the cell. Phagocytosis is a form of endocytosis where large particles like pathogens are engulfed by phagocytes, such as macrophages. The ingested material is then degraded by lysosomes. The main difference between active and passive transport is that active transport requires energy to move substances against a concentration gradient, while passive transport does not require energy and moves substances along a concentration gradient. Examples of pumps involved in active transport include the sodium-potassium pump, also known as the Na+/K+ pump, and the sodium glucose transporter, or SGLT. The sodium-potassium pump works by moving 3 sodium ions out of the cell and 2 potassium ions in using ATP. This creates an electrochemical gradient essential for maintaining cellular functions like nerve impulse transmission. ATP provides the energy needed for the conformational changes in the sodium-potassium pump, allowing ions to move against their gradients. The secondary active transport mechanism of the sodium-glucose transporter uses the sodium gradient created by the sodium-potassium pump to co-transport glucose into cells against its concentration gradient without directly using ATP. The electrochemical gradient, maintained by the sodium-potassium pump, leads to a net positive charge outside the cell and a net negative charge inside, creating the resting membrane potential. Oral rehydration solution, or ORS, is a mixture of sugar, salt and water used to treat dehydration. The basic recipe is 6 teaspoons of sugar, half a teaspoon of salt and 1 liter of water. ORS is effective in treating dehydration due to diarrhea because it replaces lost fluids and electrolytes. The glucose in ORS helps the body absorb sodium and water more efficiently, restoring hydration. The composition of ORS enhances water retention and absorption in the body. The glucose in ORS facilitates the uptake of sodium, which in turn promotes water absorption in the intestines, improving hydration rapidly.